this special section that tackles infection prevention, a topic of great relevance nowadays. It is a great pleasure to introduce you our moderator, Mrs. Elena Engqvist, an internationally experienced professional whose professional career spanned more than 35 years with core competencies in nonwovens, hygiene, and healthcare products, R&D, consumer insights, and global recognized brands. I already spoke too much, so without further ado, I will give the floor to Elena. Thank you very much for your attention.
Okay, I welcome all of you to this seminar. It's going to be very, very exciting in these times to talk about infection prevention. And it's going to be an exciting time. We're going to have many presentations. And now I would welcome Professor Dr. Didier Pittet from the University Hospital here in Geneva, and also starting already in 1994 the issues with clean hands, and going on in 2005, working then on the same thing with WHO and getting those assignments. And it's very important. And you also are a hospital epidemiologist and leading the infection control program, and today have a lot of awards from different areas and from the Queen of England, among other things. And then also there is today, it's the Clean Hospital Day, the first time it is online as a virtual event. So that means that when we finish here, you will go back to Geneva, to your hospital, and start this very important session. So you all are welcome, and the speakers will continue then as time goes on. Okay.
Let's start from there.
Yeah.
Thank you very much, and thank you for the introduction. And it's a pleasure for me to be here. And yes, it's a very specific day, and we'll come back on this very specific day a little later in my talk. So, healthcare-associated infections, a worldwide pandemic. You probably heard about this worldwide pandemic, but I will come back on some of the issue. I'd like to start with this slide, which is welcome to the hospital. Infections will be waiting for you at the entrance door of the hospitals, and unfortunately will complicate your hospital stay. This is a very old problem, and I won't come back on the history of healthcare-associated infections today, but I can tell you that at least 500,000 patients every day in the world, in hospitals only, are actually developing healthcare-associated infections. Think about it, 500,000 patients every day in the world, and in hospitals only. And I usually say in hospitals only because we don't have the statistics for healthcare-associated infections acquired outside of the hospital. I won't detail it today, but they may happen in every practice where care to patients is provided. Now, these numbers are big. They are really, really big. 500,000 patients every day, and for a total of 16 million deaths every year in the world. Think about it, 16 million deaths. It's more than AIDS, tuberculosis, and malaria together that hardly make more than 3 or 4 million deaths. So it's a lot more. It makes it the first cause of diseases and deaths in the so-called developing countries, and the second cause in our countries, the so-called developed countries. I say so-called because sometimes in some hospitals, even in this country or in the United States or in Hong Kong, you can see things that you should never see. And we'll come back on these. Now I'd like to show this video, if you can start the video. And you see the title of the video, The Daily Impact of Hospital Infections in the United States. So in the United States, the country where the most money, speaking of GDP, is given to the healthcare systems, every day there is the equivalent of a jumbo 747 crash just because of healthcare associated infections. Big numbers, very big numbers, but despite of it, nobody speaks about it. That's why I used to use these terms for the past 25 years, and I call it a silent pandemic. Nowadays, everybody knows what is a pandemic, right? At the time I was using pandemic, it was not clear to people what it meant. Today, it's very clear. We all know what is a pandemic. But in this case, this is a silent pandemic. It used to be that nobody spoke about it. When I used to start to speak about healthcare-associated infections, my CEO here at the University of Geneva Hospital said, don't speak about it too much, okay? Because it was linked to the quality of patient care, and it would mean that the hospital would be dangerous, right? But now we know that, yes, hospitals are dangerous all over the world, as well as clinical care practices. Your private physician could be dangerous for you when he is not using the perfect way to take care of yourself. Now, let's come back on these. We started, as Elena addressed it a little bit earlier, to work on hand hygiene from the early 90s. And actually, in 2005, the World Health Organization asked us to start this worldwide Clean Hands project. So Clean Hands, where did we start? What did we do? Where are we today? Where do we go? So where did we start? We started with a very, very, very simple study that told you that when people had to wash their hands less than 4 times over 10 times, they used to wash their hands, right? Which is terrible when we speak about healthcare workers, nurses, doctors in hospitals. But what we understood very clearly is that people technically had no time to wash their hands. And that's the reason why we say no hospital, no country, no healthcare system in the world can claim to have solved the problem of healthcare-associated infection and to find a solution to at least make sure that people could wash their hands on appropriate time using the appropriate way to do it. Now, because of this, we realized that time was the most important issue, and we proposed to switch from soap and water hand washing to alcohol-based hand rubbing, to the use of alcohol-based hand rubs to clean your hands, right? So we introduced what was called a system change, right? So proposing to use alcohol-based hand rub instead of soap and water in hospitals was a revolution. It allowed healthcare workers to clean their hands fast, more appropriately, every time they needed to do it, very quickly and with no resistance to alcohol, whereas we had resistance to soap. Now, when we introduced it in the University of Geneva in 1994 and monitored in parallel healthcare-associated infections, we obtained a drop of 50% in healthcare-associated infections in less than 4 years. This drop was associated with the saving of between 400 and 500 lives every year, in Geneva, here in this city, and also associated with huge cost savings. We are talking about between 20 and 24 millions every year that we were saving for the University of Geneva Hospital. So with this money, we could easily engage into promoting change in practices. So I used to say rub hands, it saves money, to sort of approach the cost effectiveness very quickly, but we published it in 2004, the cost effectiveness of the hand hygiene promotion strategy at the University of Geneva between 1994 and actually the year 2000. Importantly, in academic medicine, this is sometimes publish or perish, so we publish our papers in the Lancet, and this paper was a tremendous change because actually hospitals were visiting us from all over the world, countries were visiting us, they wanted to endorse the strategy that we had developed, And between 2002 and 2005, we tested the strategy in different hospitals. So at that time, what did we do? Well, very, very quickly, the World Health Organization asked us to start the campaign. So what we did is we reviewed the evidence. We developed a multimodal strategy to change the behavior of healthcare workers. We developed new guidelines, implementation tools. We field tested the strategy. We allow for adaptation. We'll show you some pictures. These are actually the guidelines that we developed for WHO in 2005, 2006, together with an implementation strategy that was linked to this multimodal strategy. Because when you want to change behavior, it's not enough to tell people to use alcohol-based hand rub, but you need to tell them when to use it, how to use it, to have reminders, to monitor their practices, feedback their compliance, and so on, and to implement institutional safety climate that was so important at the time. We developed these tools, which is my 5 moment for hand hygiene that has been used all over the world, which is to say, when is it that healthcare workers need to clean their hands? And this tool was translated in multiple languages, but also adapted. Here you see it in Australia, it's the 5 moment in blue. You see it in Korea. You see it soon in Argentina. Playmobil in Argentina is promoting hand hygiene. In Germany, Olaf is promoting hand hygiene. And in Japan, Hello Kitty is promoting hand hygiene. This is very important because you let people adapt. If you want people to adopt a new strategy, you should let them adapt the strategy to their culture, to their resources, and whatsoever. For those interested, I gave a TED talk on the topic that you can access. Now, here is the when to clean your hands. Here is the how to clean your hands. And these tools have been implemented all over the world, tested, adapted, so that people could use them very largely, from very modern healthcare settings to settings with very limited resources in a very multicultural environment, and also ensuring universal system change. And that's why we gave the WHO formulation to the WHO together with the recipe, making sure it could be free of patent, so it could be produced locally all over the world. And in 2008, the alcohol-based hand rub became part of the WHO Essentials Medicines list. And you have seen that during COVID, people could produce alcohol-based hand rub all over the place, here in Geneva at Firmenich and at Givaudan, as well as all over the world. It was produced for free. Now, then we developed other campaigns, the promotion campaigns. We obtained endorsement, monitoring the levels, trainer-trainer program, driving excellence, and so on and so on. We started the program in October 2005 from WHO headquarters. Here you are at WHO, the previous EB room that has been now refurnished, but still is the same. And now we are actually reaching with this campaign more than 98% of the world population. So it means we are all over the world with this campaign. That doesn't mean that we are currently in every single hospital. So it means that the campaign should continue, but at least we probably are saving between 5 and 8 million lives every year in the world with these campaigns. Now, what is important is to monitor the levels, to tell the hospitals how good they are and how they could continue to improve. So to monitor practice and ensure sustainability of the solution worldwide, we developed a tool which is a hand hygiene self-assessment form, where every hospital in the world can monitor the level of the hospital to promote hand hygiene. So it has been translated in many different languages. It has been tested very widely, and here is the result for the United States. For a large number of hospitals during the first, you see the median component for the US facilities participating in the first survey that was actually in 2011, so 10 years ago. You see system change, easy to implement in the United States, right? You just have to buy alcohol, put it at the bedside. But what is a lot less easy to implement is training and education, evaluation and feedback, reminders, and safety culture. You see, safety culture, we reach only 60%, even in the United States, as you can see here. Then we did it again in 2015 and compared it to 2011, and you see the progress worldwide in hospitals to endorse the strategy. In blue, you have the overall results, and in gray, different gray, the 6 WHO regions all around the world. Now, in 2019, if I can get the next video, please, if you can push on the video.
Hello everyone, I'm glad to welcome you again to this Save Lives, Clean Your Hands campaign, annual campaign of the World Health Organization. As you know, this campaign is engaging healthcare facilities as well as healthcare workers all over the world to pledge for hand hygiene. You can just access the website and the platform where you can enter the data so that we can generate global data for all over the world. Today we are inviting healthcare facilities around the world to submit their data online as part of the WHO Global Survey to assess hand hygiene resources, promotion and practice worldwide. By participating in this survey, you will help WHO to monitor the progress made in hand hygiene. This information can then be used to develop specific new tools that target these areas of greatest need. Please all participate to the 2019 WHO Global Survey. Improvement was obvious between 2011 and 2015. Let's see what it is in 2019. Welcome to the survey.
So we promoted in 2019, we did the study in 2020, and where are we today? Here are the results. The WHO Global Hand Hygiene Self-Assessment Survey 2019, and you see the results from all over the world. Again, system change is the easiest way to implement the strategy, but you need to have the multimodal strategy working, and you see the different level. And yes, we progressed between 2015 and 2018, but you can see that there are huge differences according to the different regions around the world. So there is still a lot of progress to make all over the world. It's very clear. Now, where do we go? How to achieve excellence in hand hygiene is the next step, and we have Hand Hygiene Excellence Award hospitals. For those interested, you access the website that we designate in order to have a snowball effect all over the world for hand hygiene. Well, we are promoting this strategy for the past more than 15 years. And where do we go with clean hospitals? This new strategy that we have been starting in 2018. Now we'll come back on these. What is Clean Hospitals? It's a coalition of international stakeholders who work explicitly to promote and support healthcare environmental hygiene, doing research, publication, participating in conferences and events like today, education and training, and specialized working groups that we have developed on specific issues. Where did we start? The healthcare environment remains a major gap in infection and prevention and control. We need to address silos and fragmentations in the field. Clean Hospitals' official launch was at Interclean Amsterdam in 2018. And for those interested, access our website, cleanhospitals.com, very easy to remember. What did we do? We brought together academia and industry to actually champion the science, the best practices, and evidence-based solutions. We created a network where projects members can actually exchange and collaborate. We created a platform from which to bring healthcare environmental hygiene into the spotlight, which is very, very important. There are too many hospitals where the CEO are reducing the resources to maintain the environment clean. In 2020, despite a very busy year against COVID, here is what we did. A lot of activities that we did. I didn't want to enter into the details, but this is a huge activity for 2020, and the forecast for 2021 will be even larger, and for 2022, I can't imagine how big. For those interested in publication in the last 2 years, they are summarized here. You can find them easily on our website in these 2 slides. The Clean Hospital Day we started last year on the 20th of October 2020. We had a specific focus on COVID-19 that was obvious. We had videos, a teleclass that met almost 100,000 people, webinars, companies were involved. We had social media activities. And today is the 20th of October, 2021. And I say, please join us. And where are we today? Actually, we are continuing our research and academic work. We are expanding our network and connections with industry. We are finding new ways to champion healthcare, environmental hygiene, and patient safety. And if we speak of today, today is the 20th of October. So I'm very happy and proud to be at Index 20 on precisely this 20th of October, because that's the Clean Hospitals Day 2021. We are today raising awareness, spreading the word, educating, and so on and so on. In particular, today is this lecture and then this discussion around different activities here at Index 20. Then we'll have the very first Clean Hospitals Digital Day. So everything will be digital. I will show you the program. And tonight I will be giving the Weber teleclass that will probably be reached by more than 100,000 people from all over the world using a very cheap system that you can even follow the lecture on your mobile in Africa. So we are developing new posters in order to engage healthcare workers and people working in hospitals in environmental hygiene into this strategy, including the concept of the Signer cycle adapted to the different type of cleaning. And you can follow up on Twitter using our Twitter account Clean Hospitals, as well as on Facebook, Instagram, and on YouTube channel. The Digital Day is an interesting concept where we encourage all of you to participate. This is a program. It will start at 1:30, and I will need to be in my office at that time for the opening lecture. Then there will be activities against cleaning and disinfection in healthcare setting. We'll discuss virucidal activities. Some companies will be presenting their own product or organizing their own symposia. And then we'll have a roundtable discussion about COVID and healthcare environmental hygiene perspectives for the future. For those interested, here is the program of today. You can access the website, go to cleanhospitals.com and access these activities and participate. All the recording will be available up to the end of this year, so the 31st of December 2021. So you can even sort of access later the lectures if you have no time to do it. There will be a symposium on actually hand hygiene, covered by the Saraya company. And the second Clean Hospitals Digital Day will be on the 7th of December 2021. You have here the lessons from the REACH study, the famous REACH study that we will be discussing with the authors of the study. We have the air treatment and purification as a topic and a panel discussion. Of course, it's a preliminary program, but that will be an exciting program. Where do we go? We continue the research and publication, academic and white papers in peer-reviewed journals. We develop, test, validate, and diffuse the healthcare environmental hygiene self-assessment framework. Remember the framework I showed you for hand hygiene? We are currently developing the same type of framework for actually environmental hygiene. Together with many institutions from around the world, and we have preliminary result of this validation. We have conducted a systematic review of the efficacy of healthcare environmental hygiene intervention on healthcare-associated infection. We'll present early data during this digital day today. We are organizing the Clean Hospitals Day, as I told you, and the Clean Hospitals Digital Day on the 20th of October today and on the 7th of December. So where do we go with this environmental hygiene self-assessment framework? The same way we did it for Clean Hands, we want to do it for actually the environment. And you can, if you access the ICPIC website abstract poster book, you can find the early results of this first actually validation test that we conducted for this new scoring system that will help every single hospital to monitor their practice in environmental health hygiene. Our current Clean Hospitals partners, you are all welcome to be part of those partners on this slide, and I want to thank them for their support and their activities and for actually bringing to us their best scientists to develop the concept that we want to develop for the future. So from clean hands to clean hospitals, I want to give you one summary slide. Clean Hands showed the way to go from 1995. Reviewing the evidence and building intervention strategies was absolutely critical. Monitoring performance, progress, and time trends ensured improvement and sustainability. So the same way Clean Hospitals was launched in 2018, the model is framed on the Clean Hospitals, on the Clean Hands model, We review the evidence, we develop strategies, we monitor practices, we ensure sustainability. So really it's the same model that we are using with somewhat the same and different partners because we are endorsing more activities into the system. And with this, I would like to thank you for your attention. Thank you. We have time for a few questions, and then we'll move to the moderation of the next part.
Well, I have something I'm very
Sure.
interested in, because using the modern technologies also, using sensors to make sure that people really in the hospital have cleaned their hands.
Yes.
Do you have some progress in that such a
Oh
thing?
yeah, we did a lot of progress, but today it's mostly at the still mostly at the level of research. There are many tools that have been developed that are currently not helping so much to change the practice, such as you enter into the room and there is a reminder, please clean your hands. It works for 3 days, 4 days, 1 month, but no more. Now today we have systems that you can carry on yourself that recognizes you and that can even recognize you when you clean your hands and if you do it correctly, and can even give you a feedback on the way you clean your hands. Those systems, of course, are part of research, but there is a good chance that these systems could be implemented soon in practice, at least in some part of the hospitals or part of hospitals, or at least in a training and sort of an education process at the beginning of some new position and new jobs. We still need to fine-tune a few elements of the strategy, but definitely in the future it will be help. I know at least one operating theater, but I know it has been implemented in several. I know one operating theater where the surgeon and others need to enter the room, clean their hands appropriately, and only when the hands are cleaned appropriately, the door is opening. So I think these are good progress, and these are things that in healthcare we need to understand how to live with.
Thank you so much. I think you're very right on this. The final one is very interesting.
Mm-hmm. It's a good example.
No, there is one question. We have to get a microphone. Someone will come with a microphone for you.
I have been the 3 questions, but I'm sure I'm not the only one.
Go ahead.
Okay. 2 simple questions and one a bit more complex. What do you think of the fact that in a country like Switzerland, a symbol of hygiene, a liter of alcohol, medical alcohol, I think you call it ethanol, costs $30 per liter?
Yeah.
There are some rationale behind that because the government does not want people to have the citizen drink alcohol instead of whiskey and vodka, but still it is dissuasive for the use of alcohol as a disinfectant by the man or woman industry.
You are perfectly right, and actually I can do the comparison with Uganda, where in Uganda we are locally producing alcohol-based hand rub solution starting from sugar can that you can squeeze to produce sugar in one side, and with the leftover you can do alcohol. And we had to negotiate with the Ministries of Health and the Ministry of Economy, saying that this alcohol could be made to produce Johnny Walker, and I understand that there will be taxes on these, but on the medical alcohol you should not have taxes. And that's what we have to fight for, because actually alcohol-based hand rub solutions, I think, are entering into our life now, in particular after COVID, and they should be free. There should be free access to those solutions anywhere, wherever, and whenever you need to clean your hands. So there is a way to go, including in Switzerland. You are perfectly right.
My second question is that since the pandemic started, every second time you ask a health professional, she would say you have to use alcohol. Every other second time they say washing hands with soap is just as effective. So it triggers, it develops a certain haziness in our understanding
Yes.
of the mechanism of
Yeah.
contagion.
That's a good question. Actually, washing your hands appropriately for COVID-19 is okay. You will remove COVID-19 from your hands. You won't kill the virus, because with alcohol you kill the virus. With soap and water you just sort of move the virus from your hands. Now, if you have the time to wash your hands, that's okay. If you don't have the time, you have no choice, you use alcohol. Now, alcohol is a lot more powerful, because with soap and water, you reduce only 5 logs from your hands. With alcohol, you reduce 5 to 6 to 7 logs. So, well, I'm not telling that alcohol is the only way to go. If you wash your hands, that's fine. The most important is to clean your hands. If you have no capacity to wash your hands, no time, you don't want to use too much soap, you can use alcohol-based hand rub. It's a lot more efficient, it's a lot more effective, it's faster, and you can make it available on your site all the time. So it's a lot more convenient. For healthcare, it's a different issue, because in healthcare, people have no time to wash their hands. We have demonstrated it. And they need, the hands need to be perfectly clean. And it's a lot, the safety is a lot higher with alcohol than with soap and water.
And my third question, I'll try to make it short. In the early years of your campaign, you had trouble with the unions of nurses. For it was more a matter of beauty of the hands and etc.
Yeah.
But then they developed this concept that nurses had the right to challenge the surgeon in the operation room, and they should be protected against retaliation if they tell the surgeon, you have not cleaned your hands properly, you should. And this opens a lot of probably side issue when you challenge hierarchy, even for good reasons. It might have a lot of side effects which you do not predict. So what is your attitude?
Well, empowerment, that's a very good question. Well, we had the difficulties with nurses unions at the very beginning in the year 2000, because we actually suppressed false nails in the guidelines that we wrote for WHO, because false nails are associated with outbreaks in hospitals and deadly outbreaks. So it was a fight with the unions that lasted for 2 years, but nowadays you don't find false nails anymore in healthcare, which is good. So now the next issue is whether you could empower people in a hospital to make sure everyone is going in the same direction. So in an operating theater, it's very important that everyone could be empowered to change the behavior of people around them. So I'm not against the principle, but it needs to be done appropriately, and it needs to be done in an institution where safety culture is really deeply impregnated into the culture of the institution, because that's the only way it would work. Other than this, as you said, it could be associated with counter reactions, and that sometimes are toxic. But most of the time when we do it, it works pretty well. So I think we need to go ahead, provide people the tools to be empowered for all of us to do a better job at treating patients in a safety way.
We have a question here.
I have one very short question. What is the proper percentage of alcohol in alcohol-based hand rubs?
Yeah, it's a good question. The proper percentage when we delivered and gave the alcohol-based hand rub formulation to the World Health Organization is that it depends the type of alcohol you are using. If you use ethanol-based alcohol, it should be at least 80%, okay? And then it depends what you use as an emollient to protect your hands. Because if you use a lot of glycerin, you need to use a little bit more alcohol in order for the alcohol to be reached by hands efficiently. If you use isopropyl alcohol, it's around 75% alcohol final that you need to use. Now, if you look at product on the market that meet the norms, you will find some product that have 65% alcohol. Now, what you have to understand is that those products usually have been tested and they meet the norms. So they are sometimes using a combination of 2 different alcohols or 3 different alcohols. So sometimes it's difficult only from the formulation to tell if the product is perfectly efficient or not. Now, if you find a product that is 40% alcohol, it doesn't work, right? If this is less than 60%, it usually doesn't work as well as needed to clean your hands perfectly.
Thank you very much.
Do we have some more questions? Yes?
Hello.
Hello.
So maybe a naive one. How can we be sure that there will be no resistance development against alcohol?
It's not a naive question. It's a very good question, actually. The reason is, and you are very right to mention resistance, because bacteria, viruses, parasites, they have this capacity to develop resistance against antibiotics, antiviral drugs, and antiparasitics, right? So technically, bacteria, viruses, parasites are able to develop resistance. We have seen it for antibiotics and bacteria. We have seen it for viruses. Look at AIDS virus that is able to switch very quickly and be resistant to any type of antiviral drugs. We have seen it for malaria. We have seen it. Now, the reason is that the mechanisms of actions are quite specific. So antibiotics have many different mechanisms of actions, but they are specific. They are targeting different specificities. Antiviral drugs, the same. Antiparasitic drug, the same. Alcohol has no specific activity. Alcohol is just exploding the membrane. So it's totally non-specific. So there is no mechanism of resistance. That's why there is no resistance to alcohol. So you will read that there may be something that is called tolerance. But tolerance is only if you give 10 or 20% alcohol to a bacteria. So then you need a lot more alcohol to make the bacteria be sensitive. But nobody will use 20% alcohol in a solution that is used for hand rub, right? I told you 60%. So that's why there is no resistance. Now there is one exception, which is not a resistance, which is alcohol does not kill spora, like Clostridium difficile spores, right? But nothing kills spores that you can add on your hands. You need to wash your hands. In our institution, actually, we don't stop using alcohol because we can control Clostridium difficile with good infection control practices, so that's not an issue. But that's a very important question that you are asking, because that's also the success of alcohol, the absence of resistance. So the capacity to resist to a totally non-specific action does not exist yet.
Actually, I do have another question.
Yes, go ahead.
Yeah, and that relates to elderly care. Coming from Sweden, you know we have had a big problem with this.
Yes, during
and
COVID.
I wanted to ask you then, how would you see elderly care should be managed better in terms of preventing the infection distribution?
Yeah. Well, in Sweden, it has been magnified at the time of COVID because some wrong decisions were made. We should say that, and that's the reason why the Prime Minister finally will quit his job, right?
Oh.
Well, in large part. Now, this was just a projector on the problem, because if you go anywhere in elderly care facilities, you will find that the quality of patient care most of the time is absolutely not the quality of patient care you are asking for in hospitals. Of course, there are always exceptions. There are very bad hospitals and very good elderly healthcare facilities. But the problem is that we don't pay as much attention. So it means we don't have infection control nurses in large amount for elderly. We don't have specific ID doctors taking care of the elderly. We don't have, and in the mission that I had the privilege to conduct for the President Emmanuel Macron, we looked at elderly care facilities in Germany, in Belgium, in Sweden, in France, in Switzerland, in Italy. And in every country, the quality and the intensity or the capacity to provide optimal care to the elderly is reduced as to compare to the non-elderly. And elderly care facility should really completely transform their attitude and their practice, in order to actually make sure that we can provide to the elderly the same type of good quality care that we are providing to anybody. And yes, Sweden was a very peculiar situation during COVID, but there are many places around the world where actually patients had suffered in elderly care facilities, including in Switzerland during the second wave of COVID, where the elderly were not protected optimally, including in many countries in Europe where, if you remember at the beginning of COVID, there was no mask for the elderly care facilities, which is a nonsense, right? A total nonsense, considering the level of acuity and the danger of the COVID-19 for those elderly. So there is a large room of progress. This Clean Hospitals movement and initiative that we are developing is, of course, covering also for elderly care facilities, because we all know that not only we will go to these elderly facilities maybe one day, but we all know that we want to provide good care to our elderly.
I think you're so right. If you could start initiating something, go on with that for the elderly care. I think it's very, very important. Thank you very, very much.
My pleasure. Should we move to the...
Okay, we will now continue a little bit with our program. And actually, Gert Riesen comes from 3M, has a high position in infection prevention. And it's going to be interesting because you have a background also from the real world in the nursing to the R&D and to the company perspective to look at the details. Welcome.
Thank you. Thank you, Elena. Thank you to Edana for the invitation. It's a pleasure being here. It's a pleasure talking about something what I also call infection prevention. And if I talk about infection prevention, I take it broadly. Normally, infection prevention measures are focusing on patients. And that's also what I do, what I'd like to do, and what I very much enjoyed in Professor Pittet's presentation. But I like to take it a little bit broader. Infection prevention for healthcare workers. And if we talk about infection prevention for healthcare workers, we are talking about occupational hazards. We are talking about personal protection. Maybe first a disclaimer. Disclaimer 1, I work in medical industry, as Helena said. Disclaimer number 2, an important one, is that it will be impossible to cover the complete personal protection, respiratory protection in 20 minutes. I can talk 2 days about it and not cover it all most probably, but I will try to do my best to cover as much as possible in the next 20 minutes. If it is about personal protection, then we have laws in place in Europe. I'm from the Netherlands, and we have laws in place, and we take them from Europe. And those laws are there for basically everybody. And many, in my travels around the world, visiting, talking to healthcare workers do not always realize that there are laws there to protect them. If I talked, and that was 2 weeks ago, I talked to OR nurses in the Netherlands, and they think that there are laws in place. They know that there are laws in place, but they think that those laws are solely for industry and for building and for that type of activities. And that is simply not true. I've here 2 directives, European directives. on personal protection, and they are saying exactly where these laws are made for. You have to know, and most of you will know, that if a directive uses the word shall, then there is no way around it. You just have to do it, period. No compromise. Now, the first directive, and that is the Directive for Safety and Health of Workers, workers in general, all workers, also healthcare workers, it says that the directive shall apply to all sectors, so also to healthcare, also to nurses, also to doctors, also to everyone working in whatever type of institution. Then the law also says that employers, so hospitals or hospital trusts in our case, they have a duty to ensure safety and health of workers in every aspect related to their work, in every aspect. In order to get a good view for the hospital, the employer, to where the hazards are for the employers, the law also says that the employer, the hospital, the trust, has to do an assessment of risks. And based on that risk, the hospital has to provide adequate PPE, adequate personal protective equipment. Now, in my travels, I talk to hospitals very often. And then hospitals, they ask me the question very often, what type of PPE do I have to use for this or that? And my answer most of the time is, I don't know, because I did not do the assessment. You have to do the assessment, and based on the assessment, you have to provide adequate protective equipment. Now, in those cases, by the way, I try to find an answer to that question with hospitals. But if you look at the letter of the law, it is the obligation of the employer. And then there is another directive, and that directive is a directive on biological agents. And based on that directive, microorganisms or hazards are divided into different groups. Group number 1 microorganisms are very unlikely to cause any disease. That are partially also microorganisms which we really need to survive. In a world without microorganisms, we would not be able to survive. We need microorganisms also. But these microorganisms in group 1 are very unlikely to cause a disease. Then you have the type 2 microorganisms. They might be a hazard to workers, but they do not easily spread, and there are preventative measures available, or there are curative measures available. So they are a little bit more hazardous, but not that hazardous. Then you have the group 3 microorganisms, and they can cause severe human disease. They present a serious hazard to healthcare workers, and usually there is an effective prophylaxis. The European Union has decided that COVID-19 is a group 3 microorganism. The European Union, and that is what I read yesterday in a white paper which was sent to me, and that was a very interesting one, has also decided that COVID-19 amongst healthcare workers is an occupational disease, is now an official occupational disease. It means COVID-19 is related to the job of people working in hospitals. And then you have group 4 microorganisms, and they can cause very severe human disease, and they spread easily into the community, and there is usually no effective prophylaxis or therapy available. Ebola is an example of this. Now, we already talked about COVID-19, and these are data from September 25, I think, yeah, September 25, for 7 countries. And in those 7 countries, COVID-19 caused already more than 2 million deaths. 2 million deaths just in these 7 countries. If you look at worldwide, it is immense. COVID-19 killed a huge number of people. And amongst them also healthcare workers. We already talked about them. And Mr. Tedros from the WHO, he confirmed that at least 115,000 healthcare workers were killed because of COVID. That is a statement also on the ICN website, and it's a statement from September. So that is where we can see that there is a real threat for healthcare workers to be to get COVID and to die because of COVID. It is an occupational disease. So now about transmission of these microorganisms. Many people think that microorganisms are floating into the air. And that is true, but they are not floating like what we call naked microorganisms. In order to be transported from A to B in air, microorganisms have to attach to particles. And those particles can be saliva, can be sputum, can be blood, can be everything. But they have to attach to a particle. Particles, the size of those particles depends on the process which is generating the particle. If I have in an operating room in ENT surgery, for instance, if I have a diathermia generator, an electric knife, then that will generate plume, it will generate smoke, and then I have very small sub-micron size particles. If I am working in orthopedic surgery and I am cleaning an acetabulum in hip surgery, then that drill is slowly moving and it is generating different particles, bigger particles. So the size of the particle is depending on the process which generates the particle. If I have a very high-speed drill in dentistry, then the particles are also smaller, much smaller. For filtering out microorganisms out of the air, it is important to know that, you know, it is what people are wearing, those masks, and some people in the audience are wearing masks. Those masks are particle filters. And what we try to do is to filter the particles, and with that, the microorganisms out of the exhaled air. That is what those filters are doing. Now, there are differences between aerosols, droplets, and splashes. Aerosols is a mixture of particles into a fluid, and the particles are not bigger or smaller, actually, than 5 microns. If you have a droplet, and that is what the majority of the particles which we exhale if we normal interact with people, if we do our normal things, then those droplets are much bigger. They are somewhere between 5 and 100 microns. And then we have the real splashes, and splashes are really big. They are far bigger than 100 microns. What we also know is the smaller a particle is, the deeper it will sediment, it will go into your respiratory system, into your lungs. So the smaller the particle is, the deeper it is. So what is important is that we filter out particles, and with that, microorganisms out of the air. Now, and in order to know how that works, you need to know first how air moves and how particles move when we bring them in air. And if you bring in an airstream an obstacle, and that obstacle can be a fiber from a filter what you put in front of your nose and mouth, what you need to know is how does that airstream move? And what you see typically is that if you bring an obstacle in an airstream, the air is moving around it very nicely, as it is as we see it here on this picture. Filtration is not more than bringing particles in the air in contact with the obstacle. Because if we bring particles in contact with the obstacle, the particles will attach to the fiber, in this case the obstacle, in this case the fiber. So that is what it does. Filtration is not more than bringing particles in contact with the fibers. Now, another thing is, and that is a misconception, is that people believe that filters, masks, work as a sieve. That it is easy for small particles to go through that sieve, and more difficult when the particle is getting bigger. And that is not necessarily true, because what we basically do with regards to air filtration, we define three types of particles in sizes. We have particles which are smaller than 1 micron, 0.1 micron, the sub-micron size particles. We will find them in laser smoke, diathermia smoke. We have particles somewhere between 0.1 and 1 micron. They are very difficult to be filtered out. They are the most difficult one, to be honest. We will see that later. And we have the particles which are bigger than 1 micron. But the smaller particles are very easy to be filtered out. And we will show that in this slide. Because it is not only how air moves when we bring it in contact with an obstacle, with a filter, but it is also how particles in that air move. And what you see, if you look at these very small sub-micron size particles, the smallest particles, they collide with oxygen molecules, with air molecules, continuously. And due to these collisions, they change their direction as well as their speed continuously. So they make these type of movements. These movements are called Brownian movements. And because of the Brownian movements, they will always connect with a fiber of a filter. So they are filtered out very easily. Then you have the particles between 0.1 and 1 micron. Those particles, if you look at them, they follow the airstream more or less exactly. And with that, they would not be getting into contact with the fiber. But if you bring an electrostatic charge to the filter, to the fibers, then these particles are filtered, are drawn as a magnet, drawn out of that airstream, and that is how that works. That is also the reason that masks, filters, have an expiration date. If a mask is getting wet, it will be less effective against these type of particles. And then you have the big particles. And the big particles, I have another presentation where I have a picture of myself in the car with my mother-in-law, and I didn't want to show that here now, but my mother-in-law is a big particle. It's a nice particle though. It's a very nice particle. It's a big particle. If I would put her in my car next to me and I would blindfold her and I would make a very sharp turn to the left, my mother-in-law would go straight forward in the original direction because of her mass, because of her original direction, and because of the speed. And that is also what these big particles do. If the air divides because of the obstacle, the filter fiber, but the particle goes straight ahead and is captured. So that is what we need to know in order to look at filtration. And then we have two different devices. We have surgical masks or masks, and we have respiratory protective devices. Two completely different devices, completely different. I see people sitting here in the audience with a surgical mask on. These people, they protect us, but they do not protect themselves so much. A surgical mask was for the first time introduced by Jan Mikulicz Radecki, a surgeon, Polish surgeon, and I think he was in Frankfurt. And he listened very well to the stories from Pasteur and the stories from Lister. And he said, my goodness, we have microorganisms, that is one. Second, those microorganisms, they can cause disease, that is Pasteur who said that. But those microorganisms are also in our nose, mouth, throat. So we have to prevent that those microorganisms get into the surgical wound. And that is when he started to make and use his own surgical masks. And that was in the early 1800s, I think it was. Now, from the very beginning, surgical masks were there, and that is still the case. From the very beginning, surgical masks were there to protect the patient, to protect the aseptic environment. They were not there to protect the wearer of the mask. Surgical masks only work from inside out. They do not work from outside in. Now, but how can a mask, if you wear a mask, and this is a previous colleague from me, Karin Slootjes, for the people who know her, if you wear a mask like Karin does here on this picture, you will feel if you exhale, air is coming out on all sides of the mask. Now, we already saw that particles move in air. How can a mask like this protect a patient or an aseptic environment? And the answer to that is very simple. Because of the fact that big particles, and that is what we exhale normally, big particles, they have their own speed, their own direction, and their own velocity. And they will move ahead, straight ahead, into the filter, while the air is moving up, down, and to the sides. So the air escaping from the edges of a mask is clean from those particles. Now, the reason that a surgical mask does not protect the person who is wearing it is because of this. The seal between the mask and the face is not existing. If you inhale and you have a mask in front of your face, air has two options. The air is either going through a filter or the air is going to the sides of that mask. I am also a teacher, and I sometimes compare air streams with students. They always take the way of least resistance. And that is what air does, that is what these particles do. So the particles in the outside air, they enter the inside of the mask through the sides, because there is no seal there. Now, there is a standard, a European standard, EN 14683, and I'm sharing that group of people, and they test surgical masks. And surgical masks, we have two types, type 1, type 2 masks. Since the last revision of that standard, type 1 surgical masks should not be used to protect patients in hospitals, in operating rooms anymore. The standard is actually very clear. It says even now that type 1 masks should only be used in endemic or pandemic situations to protect the environment from contaminated particles. So that is what EN 14683 does in type 1. And type 2 masks are the surgical masks. Now, let's move a little bit ahead. If you need to be protected yourself, if you want to be protected, then you need a different device. And that device, what you need in that case, is a respiratory protective device. And that respiratory protective device works from the outside in, just in the other direction. And it works to prevent, it prevents you from contracting a disease because there is a perfect seal between the face of the person who is wearing it and the mask. Particles in the air are forced to go through the filter, and they will be filtered out there on that way. Now, that also means that you need to have a super duper perfect seal between the mask and the face. What I see very often, if I look at television and look at the actualities, this is what you see in many hospitals. People want to protect themselves, but they did not shave, and especially the males in that case, of course. If you want to be protected, then you need to have a clean shave. Even a very short stubble already jeopardizes that. Oops, oh, sorry for this. Already a very small stubble will jeopardize the effectivity, the efficiency of a respiratory protective device. Respiratory protective devices are tested for total inward leakage, the number of particles, the efficiency of the total device. And if we talk about total inward leakage, we are measuring three things. We are measuring the efficiency of the facial seal, we are measuring the efficiency of the filter, and we are measuring the efficiency and the effectivity of an exhalation valve if it is present. So, and all those three components, they decide how big the total inward leakage is. We have three types of respiratory protective devices. We have unvalved ones, so that is what this is. This is what you see very often, and I even see that someone in the audience is wearing them. They are the unvalved respirators. In many cases, at least for the company which is shown here on this behind this logo, the unvalved respirators are also tested in accordance with Medical Device Directive EN 14683. So they are dual, they have a dual CE certificate. They can be used to protect the wearer of the mask, as well as they can be used to protect the patient, the wound, or the aseptic environment. This is a valved respirator. Some people think that this thing, what is on top of that respirator here, that it is an extra filter. It is not. It is not. It is an exhalation valve, and if you exhale, that valve opens, air goes out easily, it has less heat build up on the inside of the mask, and if you inhale, if you need protection, the valve is closing. So then air is forced to go through the filter. Now, this is a more comfortable respirator, but it cannot be used in cases where you need to protect a patient or an environment. And therefore, we have also shrouded valve where the valve, the exhalation valve, is covered with an extra filter. So those valved respirators should not be used in operating theaters, for instance. Now, total inward leakage, we talked about it. We have FFP1, FFP2, and FFP3 respirators. FFP3 is the best one, has the lower total inward leakage. This is the last slide. If you look at how can you identify masks from respirators, simple, by looking at the packaging. If you look at a surgical mask, on the packaging, only on the packaging, not on the products, you will find a CE mark, the number of the standard, and on the packaging type 1, type 2, or type 2R. If you look at the respiratory protective devices, you will have it not only on the packaging, you also need to have those items mentioned on the product itself. So you have on the product and on the packaging the CE mark, the number of the standard EN149 in that case, the FFP class 1, 2, or 3, a 4-digit number from the notified body, and the NR or R devices. Now that is, let's say, my presentation for you. As I said, it is impossible to cover personal protection, respiratory protection in 20 minutes, but I would be happy if we have time for a question still.
Actually, what we will do is, after all the presentations, we will have time for questions. So please write down your questions now, so we can have everything for the discussion. There will be plenty of time then. Okay?
And I will be walking around.
Thank you. Yeah. So now we're going to introduce Adrian Fellows from Gamma Healthcare, Technical Director and well known in the business with everything that relates to good hand hygiene, disinfection, infection control. Welcome.
Thank you, Helena, and thank you to the IDANA committee for inviting me to give this talk. It's particularly important for me, or of value to me. I've attended every index since it started in Amsterdam 1,000 years ago. We've seen a transformation in this industry, and nowhere has that transformation been more greater than in the areas of hygiene and infection control. Now, where's it? Is this rolling? Yeah, okay, I think we've got it now. So, the topic today is hand hygiene, disinfection, and infection control in a time of pandemic. So it's worth looking at what is the pandemic. This particular pandemic was an unrecognized respiratory virus first seen in Wuhan, China. There may still be some debate as to what the real origin was. It's almost certainly a zoonotic infection. It probably came from bats. What the rest of the route was, we'll probably have to leave to the politicians. So SARS-CoV-2, it's an enveloped virus, enveloped coronavirus, with spikes of antigenic protein embedded in a lipid layer, which rapidly spread around the world. Some people would, well, many people would agree that the enveloped viruses are relatively easy to kill, which this led to an emphasis on soap and water in the beginning, which probably wasn't helpful. It's a virus with a very large genome, as viruses go. As a result, it's capable of mutation to numerous variants, and we've seen a few: alpha, beta, delta, and probably beyond. I think there's a lambda in Peru at the moment. The genome, I think, is about 30,000 units. So depending on your view of statistics, that's sort of factorial 30,000 potential mutations. It's a respiratory mucosal pathogen spread by droplet infection. Now, you've heard from both the previous speakers that yeah, droplets come in various sizes. So droplet infection would be the droplets that fall to the ground within the sort of 2 meter range. There actually was more than enough data available from research into the common cold in the past to know that the particles would actually be over a much wider range, including aerosol particles. The 2 meter advice was a little bit premature. It initially colonizes the nasopharynx of the mucosa, and there's a lot of published evidence that it resides there for a short time before it moves down the respiratory tract. So that is an area for intervention. And one of the reasons why, for instance, masks have been are now seen to be so ubiquitous. It's primarily an airborne infection. However, it has a significant route of infection by transfer from droplet contaminated surfaces and equipment. This is most important in hospital and care settings where the viral loads are high and where the workers and the patients are exposed to significant dose escalation, which you don't see walking up and down the street. Also, obviously, you've got vulnerable people in those situations. Interestingly, there are papers that have been published. Weber in the States has published a paper showing or tracking the spread of an infection or coronavirus infection from a contaminated lift button. So even in the general environment, we do need to be careful. But it is high-touch surfaces, and there is no benefit in people disinfecting chairs and other items to the level that they're so sticky people don't use them. We are here at Index, so I'm focusing on the role of nonwovens in the pandemic. Disinfectant wipes, both for surfaces and skin, have significant roles to play in preventing spread. These typically use Thermabond, Spunlace, Spunbond, nonwovens. Obviously PPE and masks and suits that we've heard a little bit about already. These provide barrier protection, largely Spunbond nonwoven. Isolation units, where nonwoven is used in the structural wall, the roof, the barrier, to protect the patient and isolate the patient from the hospital or from the environment. It may not necessarily be in a hospital. Usually, again, high weight Spunbond. So, masks seem to have become ubiquitous. We've managed to find one with Geneva on it. Do they really work? I mean, masks do play a part when you've got a pandemic that is not only present in the healthcare environment, although as the previous speakers have pointed out, the rates of serious infection and death are much higher in the healthcare environment. This was a paper published in Nature at the end of September, showing that even in a relatively low and middle income country, the wearing of masks had a role to play. So how do we, as an industry and as individuals, what are the chances of, or what are the opportunities for breaking the chain of infection? I'm not going to talk a great deal about this because you've heard from a more knowledgeable speaker and in greater detail about the difference between masks. It is worth saying and that surgical face masks in the environment generally, or in the population generally, have another important role to play. Not only do they have a role in preventing you spreading an infection, they are important in that they prevent you getting your own hand and fingers in contact with your nose and mouth. And that's a significant role for the face masks. There's ample published papers to show that in a healthcare environment, the higher grade masks provide significantly more protection than the surgical face masks. Face shields we've seen, they have a role to play, and a lot of, in healthcare environments, you'll see the worn in combination with masks. Certainly in primary care, you know, patients like to see their doctor's faces. They don't really want to, children particularly, don't like to be attended by a masked individual. This is a new product, powered air purifying respirator, designed to overcome many of the drawbacks of some previous technologies. Was developed or is being developed in combination with the University of Manchester. Hand hygiene we've heard a great deal about. It's crucially important. It's crucially important, as Professor Peter says, not just in the context of coronavirus, but in the context of many other infections, and particularly in hospital-acquired infections. I would just say I've been involved in this game for a long time. I actually patented a disinfectant wipe in 1972. And as late as the 1980s, I was being told that the environment plays no part in hospital-acquired infections. They're autogenous and they'll always be 10%. I was also told that by one of the two leading experts in the field at the time that disinfectant wipes will be introduced to the NHS over his dead body. It shows times change. So breaking the chain of infection with hand hygiene. What the effective there was supposed to be in significantly bolder type. Good hand hygiene through washing and the use of effective hand sanitizer plays a significant role in preventing the spread of virus particles from hand and contact, well, I should probably say, and high contact surfaces to the nose and face. Unfortunately, with the need to act fast, there have been a lot of low-entry, poor products. As Professor Peter said, you see alcohol products at below at around and below 40% alcohol. That's completely ineffective. Completely ineffective. And some of the formulations that have appeared on the market are cosmetic rather than effective. But high-touch surfaces, particularly in healthcare and social care, it's essential that they are disinfected, because in a healthcare environment, particularly in an ICU or a hospital that's dealing with coronavirus, then there is significant dose escalation in terms of much higher loads of virus than you would see in the community. So all of the the high-touch surfaces, as well as the hands and other pieces of equipment, need to be effectively disinfected. This is, in some respects, not as easy as you may think, because a lot of this equipment is classed as a medical device. So as a little disinfectant wipe may have to go through full medical device regulation approval. Another major opportunity for breaking the chain of infection is isolation. Hospitals have practiced isolation and isolation nursing for many years. It's important that we isolate the COVID-positive patients as soon and effectively as possible. And in the community at large, we now need to isolate where possible. So we have pop-up isolation rooms. These are beginning to play an increased role. They bring significant benefits. In the UK, we introduced Nightingale hospitals. This is an interesting exercise. In fact, our clinical director, who I think Didier will know, Martin Kiernan, was seconded back to the Nightingale hospitals to head up infection control for that. These hospitals were basically put up very rapidly, very competently, involving the army and other people. I think largely because it had been seen that the Chinese had built 10,000 bed hospitals in very short times. And they thought, well, if they can do it, we must do it. Unfortunately, politicians did not seem to appreciate that infection control nursing is a very, very skilled pastime, and there were no nurses for these hospitals. So most of them sat empty at great cost to the taxpayer. Some of them were repurposed for other tasks, but they've now been pretty much stood down. The pop-up ready rooms and similar isolation facilities, because they're very compact and easily rolled out, that product can be erected by one person in 5 minutes. There shouldn't be a need for Nightingale hospitals in future. It can be done nearer to. And in the community, unfortunately, we've had experienced lockdown, which has proved effective. It's not without contention around the world. I have a daughter in Australia. She's been locked down for 109 days on something like 1,000 cases in New South Wales. I think I looked at the figures yesterday. The UK is still running at something like 40,000 cases. But we are at 85% plus vaccination. Right. Quarantine. Isolation, again, from a non-woven point of view. Vaccination, obviously, is the gold standard. Here you can see one of my colleagues who, again, was volunteered and was seconded from the company for the vaccination program. Vaccination works, there's no doubt about it. We've all got a great debt to the people that have developed these vaccines so quickly. But in a vaccination facility, there's still a huge requirement for high levels of environmental disinfection and infection control, because you've got sites where the vulnerable people are mixing with the less vulnerable, people who may be carrying the virus, and staff who are possibly being exposed to high levels of dose escalation. So it's important that we have a full panoply of tools available at the vaccination sites. Briefly, I'll mention nasal decolonisation and protection. This has been pretty much neglected, but as the virus settles in the nasal mucosa, it makes sense to a lot of people that you don't just have a physical barrier external to the nose, protect the nasal mucosa. And certainly doctors in the UK are now not putting aside conventional PPE, but they're also applying nasal decolonisation measures. I always do it if I'm going anywhere crowded. Thank you very much. Any questions? Take the questions at the end, yeah?
Very much. Thank you. Welcome Dr. Matthew Tipper from NIRI, the Nonwoven's Innovation and Research Institute in the UK. And you're CEO of this organization and has a lot of background also in textile engineering. So welcome to share your insights here.
So, well, good morning, everybody. I've actually lost my voice, so hopefully they're going to play the pre-recorded version, and you can listen to me, you know, several weeks ago when I recorded it.
Problem with his throat.
Might need the sound on as well.
Today I'm going to talk to you about non-woven enabled hand hygiene devices which were designed for infection control purposes. So
that's what I sound like normally.
Hello, my name is Matthew Tipper. I'm the CEO of NIRI, the Nonwovens Innovation and Research Institute. Today I'm going to talk to you about non-woven enabled hand hygiene devices which were designed for infection control purposes. So NIRI is a globally operating company and a leader in the field of non-wovens engineering and product development. So we have over 30 scientists with over 350 years of textile science experience, and we're utilizing that knowledge to improve products, to develop new products and make them better. And we have prototyping facilities to make any type of non-woven to be able to make that possible, as well as the analytical facilities to understand what we've made so we can understand what to make better. So I'd like to start my talk today by talking about Thomas, a 52-year-old man with 5 children. He had heart problems and diabetes. The diabetes left him with broken skin, areas making him a high risk of infection. In November, Thomas had minor elective surgery. So he had a toe removed, which was related to his diabetes. He was discharged from hospital soon after, in early December. 1 week later, he was readmitted to hospital, suffering with a chest infection. It turned out he'd actually contracted MRSA whilst in hospital for his elective surgery. and he died soon after of MRSA septicemia on the 14th of December. And this is the reality of healthcare associated infections. You know, we estimate in the UK there's 28,500 patient deaths every year and many thousands more cases of HCAIs. So, you know, these could account for up to 21% of the number of all bed days in NHS hospitals in England every year. And the cost is estimated to be around £2.7 billion to the National Health Service in the UK. And you know, sites of bacterial contamination are common in hospital wards. You know, there's bacteria everywhere. They're on the machinery, they're on the hard surfaces, they're on curtains, they're on bedding. So anything we can do to prevent the transmission of this bacteria is going to massively improve safety for the patients. And it's estimated that 80% of infections are transmitted by human hand. So, you know, what areas do people touch and contaminate? Well, you know, we believe that the doors in hospitals become a major reservoir for infection. So we've developed a product we call Surface Skins. And this is an antibacterial pad that sits on the door. As you press the door open, it delivers alcohol gel or an antibacterial agent to your hand. And it's all done under the pressure of opening the door. And it's not really designed to clean your hand, you know, it's designed to complement the gel dispensers. What it is designed to do is to kill the bacteria that's on the surface of the door that you're touching, so thereby breaking the chain of infection. And this product, you know, it couldn't work without non-woven technology at the heart of it. So there's a non-woven fluid reservoir and also a mediating layer that controls the amount of gel that's delivered through a microvalved top sheet that's based on film technology. So at the heart of the technology are non-wovens. So we've done a number of clinical trials on this product. So the first one is does it actually kill bacteria? And I'm pleased to report that it's very effective at killing bacteria. So we measured it against E. coli, Salmonella, Staph aureus, amongst others. And we investigated the product on the first day of use and also after 7 days. And you know, the product can last up to 7 days. And what we found is, compared to the copper and aluminium jaw plates that are currently used, we were getting almost 100% reduction in bacteria, even after 7 days. As a secondary effect, you know, so not only does it kill bacteria, we also found that we got an increase in the use of the hand gel dispensers that are currently in hospitals, which we really weren't expecting. So the product was placed near operating theatres, and we actually saw a doubling in the increase of gel dispenser use. And we think this is related to the fact that it's a slightly wet product when you touch it, and it reminds people to use the gel dispensers. So, you know, the use of gel dispensers is pretty low. You know, it can be as low as 30%. So anything you can do to increase that can have a major impact on infection control. So I'd just like to summarize, you know, what I've covered here. So healthcare-acquired infections, you know, result in 28,500 patient deaths in the UK every year. In the US, a patient dies every 15 minutes from these infections. The CDC estimates that 80% of infections are transmitted by human hand, and Surface Skins is designed to break the chain of infection, not only in clinical environments, but also in municipal buildings, in commercial environments such as, you know, food preparation or cruise ships, anywhere where there's a chance of infection breaking out. And we've proven that the product provides a rapid bacterial kill, in seconds. So the next user of the door is not going to be, the bacteria is not going to be transferred to the next user. And it's also shown an unexpected increase in hand hygiene compliance among healthcare workers, which we believe to be a real added benefit. So if you want to talk about this technology or some of our other technologies or our non-woven development services, then please get in touch. These are my contact details. And thank you very much for listening.
Thank you.
Convinced about that. Okay, we will now recorded presentation because Jason Beard at Ahlstrom Munksjö is in the US and he could not come here. And he's leading the product development and marketing teams at this company and has a long experience, more than 20 years experience in medical supplies industry. So it's going to be interesting to hear what he's talking about. Advancement in breathable viral barriers. Okay? You have the recorded version to present.
My name is Jason Beard, and I lead product development and marketing for Ahlstrom Munksjö's medical barrier fabrics. Today we are excited to present advancements in breathable viral barriers. We hope this presentation will be useful as the world becomes more aware of the need for viral protection. Before we dive into the technology, we need to take a step back and understand the market and end user needs. Infection control is important, especially in applications like high-risk surgeries where there is an opportunity to come into contact with more fluids. There are many risks to the patient and healthcare staff. Therefore, the control of microorganisms, viruses, bacteria, and fluids is of great importance to avoiding infection in every way. Wearing surgical gowns and other medical apparel like surgical masks and gloves is of utmost importance as there will always be microorganisms present on or in the skin, even after conducting strict hygienic and surgical scrubbing procedures. Surgical gowns are considered one of the most important protective items used during surgical procedures. They play an essential role in maintaining sterile conditions by blocking the transfer of harmful microorganisms and chemicals to and from the patient and reducing the transfer of bacteria from the skin of the surgical staff to the air in the operating room. So, how is protection defined for viral-resistant personal protective equipment? There are industry standards used to test and measure the barrier performance for liquids and bloodborne pathogens. These standards include PB70 AAMI levels of protection, ASTM 1671, and ISO 16604. To be qualified as a PB70 AAMI level 4, the surgical gown must pass ASTM 1671 level of protection in the critical zones. ASTM 1671 measures the resistance of materials used in protective clothing to the penetration by bloodborne pathogens. ASTM is similar to ISO 16604, which is commonly referenced in Europe. To create a viral barrier, most PPE, including surgical gowns, use a layer of film to stop the penetration of viruses and other biohazards. Some gown designs use non-breathable film, while others leverage breathable film technology. First, we will look at the non-breathable film gown designs. There are two typical gown designs that use non-breathable film. The first gown design is zoned reinforced gowns that are made with spunbond melt-blown spunbond, or commonly known as SMS fabric, with a non-breathable film adhered to the SMS in critical zones. The benefit of this type of gown is that it is cooler since the SMS allows moisture vapor to pass through it outside the critical zones. The downside is that the viral protection is limited to where the film is placed on the gown. In contrast, impervious gowns offer 360 degrees of protection. However, since the entire gown is made of non-breathable film, it can feel significantly hotter since the moisture vapor cannot escape, creating a sauna effect. Healthcare providers' confidence in the operating room is a key component of patient safety. Various factors may compromise the performance of healthcare providers, and in particular surgeons. Heat stress being a major contributor. There are many heat stresses in the surgical setting. Heat may cause a stress response for surgeons who wear impervious surgical gowns that are not breathable, therefore trapping body heat in and raising the body temperature. For the surgeon, the difference of just a few degrees can shake concentration and cloud thinking, impacting the well-being of the patient. Responses to heat may include changes in cognitive function, which could affect memory, reaction time, and complex motor skills. The bottom line is that heat stress impacts the surgeon, therefore keeping their body temperature comfortable increases performance and allows them to focus on what is most important, the patient. One way to reduce heat stress on surgical personnel is to use breathable surgical gowns. Breathable impervious gowns are constructed with a tri-laminate fabric that is made up of either microporous or monolithic film. These gowns have a high moisture vapor transmission rate to keep surgical staff comfortable, cool, and dry during long procedures. They offer 360 degrees of viral protection without creating the sauna effect, which is created by non-breathable gowns. This is our moisture vapor transmission demonstration video. In this video, you will see how breathable and non-breathable laminates handle moisture vapor. The first fabric is a non-breathable film laminate. The non-breathable film laminate is placed over heated water. After 30 seconds, you see that no moisture passed through the fabric. Now let's see how the breathable laminate performs. As you can see, the breathable laminate allowed moisture vapor to pass through it, which allows wearers of this surgical fabric to be more comfortable. There are two common film technologies used for breathable viral barriers, microporous and monolithic film. Let's start with microporous film. This film is made with fillers, often calcium carbonate, impregnated into the film structure. Using a physical process, it is stretched to create breathability. Since the fillers do not stretch with the film, space is created, resulting in voids/pores that allow moisture vapor to pass through. The more the film is stretched, the larger the pores get. This process creates a risk for viruses to get through if the pores get too large. The next slide further illustrates microporous film.
Microporous film is made with fillers. To create breathability, the film is stretched. The fillers in the film do not stretch, therefore space is created, which results in voids or pores that allow moisture vapor to pass through. The more the film is stretched, the larger the pores get, creating a risk for viruses, bacteria, and fluids to get through.
As you think about the types of film, microporous or monolithic, take into consideration ASTM 1671 and ISO 16604. Like we discussed earlier, Both of these tests measure the resistance of materials used in protective clothing to the penetration by blood-borne pathogens. Specifically, in order to pass the test, the material pores must be smaller than the 31 nanometers. Otherwise, you run the risk of exposure to infectious diseases. However, it is important to note that viruses exist that are smaller than 31 nanometers. Multi-layer copolyester monolithic film uses a solid sheet of chemically active resin to allow for breathability. Water vapor molecules pass through the chain of chemical groups using osmosis, therefore the film is not compromised, keeping the fabric intact to provide impervious protection from even the smallest viruses known. The next slide further demonstrates multi-layer copolyester monolithic film.
Monolithic film uses a chemical process to allow for breathability, so the wearer remains comfortable, not hot and sweaty. Water vapor molecules pass through the chain of chemical groups using osmosis, so the film is not compromised, keeping the fabric intact to provide impervious protection. Monolithic film delivers impervious protection and comfort, something difficult to achieve together.
This is a synthetic blood strike-through demonstration video. In this video, you will see how monolithic and microporous film fabrics stand up when confronted with blood strike. The first fabric that is tested is a microporous fabric that is commonly available on the market. In this circumstance, the microporous laminate was not able to stop blood strike-through, potentially due to overstretching. Now let's see how the monolithic film laminate performs. As you can see, the monolithic film laminate stops synthetic blood from passing through it when challenged. Within monolithic copolyester film laminates, there have been advancements as well. The figure on the left is a cross-section of a legacy copolyester film laminate. Notice that the melting temperature of the film is considerably higher than the outer nonwovens. The heat necessary to melt the film may damage the outer nonwovens, creating a slim margin of error to work with when creating gown sleeves. It is important to note that in surgical gowns, the seam on the sleeve must be as protective as the fabric itself. As you can see on the right, innovations in film technologies has allowed the melt temperature of the copolyester film to be comparable with the melt temperature of the outer nonwovens. This gives operators a wider margin of error to create seam seals without degrading the fabric. Breathable viral barriers have long been used in surgical gowns. There is a growing awareness to have viral protection outside the operating room. This has led to new fabrics optimized for use outside the operating room for environments like laboratories and emergency medical services. These robust fabrics contain the same monolithic film used in surgical gowns, but are combined with strong outer nonwovens like bicomponent spunbond to make them appropriate for use in a wide range of applications. The table below highlights the key differences between breathable film laminates and commonly used coverall fabrics. Notice that breathable monolithic copolyester film laminates offer protection from blood and viruses that is not available in flash-spun high-density polyethylene or SMS fabrics. It also provides a higher resistance to fluid penetration. This concludes our presentation on advancements of breathable viral barriers. If you have any questions, feel free to contact us at medical@alstromungsho.com. Our website also has additional information regarding breathable viral barriers at ostromungshow.com. Thank you for your time.
He will be available later on online, most likely, for the quick Q&A session. But now we will continue with Elena Cordoblas from Barry. And you have a Master of Science degree in Materials Engineering and a long experience also in the nonwovens industry, based at Dow and some other companies. And you're welcome to talk on a different aspect of the materials for infection prevention.
Thank
Good.
you, Elena.
Yeah.
Good morning, everyone. Okay, so thanks again to Dana to invite us to speak today, and also to the other speakers. What I would like to drive you through today is the Barry vision on infection prevention. I'm trying to make this work. Let's see. Green, right?
The green, the big
Yes,
green.
the big green. Okay, it's working. So we will review the recent past, the present, and the sustainable future for infection prevention. I also would like to take the chance to share with you a few information about Barry, for those of you that do not know us. Barry, it's a global solution provider for the hygiene, healthcare, specialties, and consumer markets with nonwovens, films, and laminates, as well as packaging solutions. The people that we are here today and this week at Index, we are part of the HH&S division, which has 40 plants globally and a $2.6 billion US revenue. So when we look at the recent past, infection prevention has a very complex supply chain and a global one. Before COVID, it was working well. And after COVID, the fragility of it really raised. So before March 2020, Europe was strongly dependent on Asia, and especially China, for the supply of medical device apparel products. When COVID started, with the challenges and limitations related to it, Europe had to find alternative solutions to cope with an extreme growth in demand and the lack of supply from Asia. So if we look at the data that Schlegel & Partners shares at a previously done event, the demand growth was above 60%, and due to the lack of supply from Asia, the European production increased by 170%. This basically means that in Europe there is the capacity and there is the know-how to supply converted products. And I will go back to this in a minute. So even though at the beginning of 2021 the supply from Asia got back to normal, if we want to say like this, other challenges were on the way, like a lack of supply of raw materials, as well as very high costs for freight and for transportations. If we look at the 2020 and 2021 data, we can see that there was an unprecedented time of force majeure for chemical plants, so also our resin suppliers. This meant that polypropylene was not even available on a spot allocation. In Barry, thanks to the fact that we are also a very big buyer globally of resins, we were able to cope with this thanks to our relationships, of course, thanks to our team, which also includes our customers, and we were able not to stop the line, as well as maximize our capacity. So here, resilient, it's in the title, right, which is a word which is very heavily used during the pandemic, but I totally believe this is true. Thanks to the team that was resilient, united, and agile, again, also together with our suppliers and with our customers, we were able to support them and to work together as they one. So really a great team, and here I also include the customers and the suppliers, as I said. So if we look at infection prevention today, which are the solutions that we see in this market? We believe in a multifaceted approach to infection prevention, which basically means we support the risk protection market with coverall solutions. We participate in the surgical market with gown solutions, among others. we also have solutions for filtration, like with Synergex products. And we also think about cleaning with our wipes Chicopee brand. So when we look into coveralls, coveralls have been and are a fundamental PPE when looking into the pandemic. They had a very strong increase in demand and a big focus on EN 14126 compliance. In the middle of the pandemic, we launched the European Coverall Portfolio Solutions, which was made of off-the-shelf products produced in Europe, externally tested, to allow shorter certification time and cost, as well as shorter delivery time compared to non-European solutions. The breathable, the non-breathable, and the SMS are produced here in Europe and are off-the-shelf. If we look into gowns, I think also the previous speaker mentioned that, and as well as the other colleagues, protection is really what any healthcare workers deserves every day. And it is said that AMI level 4 is the highest level of protection for surgical gowns. With the Berry solution and with design and formulation of it, we are able to supply robust products that can combine protection with breathability and comfort. We can deliver robust seam seals, whether during converting or during the use, so talking about performance in time. These are pinhole-free, which is fundamental in terms of protection and compliance. They have superior breathability and softness with different solutions that we have in the portfolio. And they have global compliance because they comply with Europe as well as UN standards. And if we also dive into filtration, our Synergex Comfort for surgical face masks and our Synergex Pure for respiratory face mask answer the need of protection, breathability, comfort, as well as compliance and quality during time. During the pandemic, Berry was able to react with agility to the strong demand of our customers and of the market. We installed 3 new Melblon lines in the region, and we were also able to optimize our processes to increase our capacity and deliver customers based on their new need. Our products are compliant with the standard regulations, have a proven quality. And thanks to these optimization projects that we made during the pandemic, which was to deliver down-gauge material with same or higher performance versus the reference, we were also able to optimize, of course, capacity, but also overall footprint of the products. If we think, for example, about how much material you can put in a truck or in a container. And I will go back to the footprint in a minute. So something maybe new in the today, in the today's seminar, we really would like to approach the sustainable future of infection prevention, okay? I haven't heard a lot about it today, but it's on the agenda, and I would like to walk this together with you. So has any one of you seen this picture before? You can also raise your hand so we, we see if you have seen it. Maybe not, right? But most of you have seen this or similar ones, right? Healthcare accounts for 4.4% of the global CO2 emissions. 4.4, okay? This is equivalent to the annual emissions from 514 coal-fired power plants. This means that healthcare has higher CO2 emissions when compared to airline industry. So in 2019, Healthcare Without Harm issues the first ever global report where the healthcare footprint globally is measured. There are several different data, among which type of emission or where this emission coming from, as well as countries, among others. And the top 3 emitters, being Europe, China, and US, account for 55% of the CO2 emissions. This means that those of us that we are here today, those of us that are following online or virtually the index, we can really make a change. We can do something for this. Because of this, we understand that the goal is to reduce CO2 emission at global level. So in the Americas, North America, so US and Canada are really leading the way. And when we come to Europe, the NHS and UK are really setting very stretched and ambitious goals in the region, as they want NHS to become carbon neutral by 2040. They also want to support their suppliers and partners to reduce their CO2 emissions. So it's really an active plan. And when we go to the rest of Europe, thanks to the European Green Deal, the target is to reduce the CO2 emissions by 55% by 2030 and to become a carbon neutral continent by 2050. If this happens, Europe would be the first carbon neutral continent in the world by 2050. So when you look into net zero, there are several things you need to look into, right? So it's processes, plants, operations, different steps in the value chain. You can use less energy. You can change the type of energy that you use. You can change the type of raw materials that you use, so on and so forth. Keep in mind, move away from virgin fossil fuels, as I will explain this in a minute. So when we look into the IPAC 2025 and the IMPAC 2025 plus targets, these represent the targets for Berry in the EMEA region for our segment, so for our business unit HHNS, for sustainability. These are targets, and we have proven data that we are complying with this, or we are above, or we are a bit below, and you are welcome to visit us at our booth to see this. We have three main pillars: products, performance, and partners. If we look into products, we are proud to say that we have 2 plants which were ISCC Plus certified in the region this year, and this goes together with our mass balance plan. When we look into performance and processes, we moved this year all our Spain plant, whether from our segment or other segments, to solar energy. And if we look into partners, we would like to become the first non-woven company to be able to support customers with our recycling scheme. And this is thanks to external and internal partners, among which our recycling business unit. So if you want to find the right solution for a sustainable product in infection prevention, I think that a lot of doubts and potential options really pop up. What we think is that mass balance is one of the ways to go. But what is mass balance, right? I'm sure some of you are familiar, some others are not. Mass balance, it's a chain of custody. It's a bookkeeping of green credits overall the total value chain. What happens is that mass balance certified products, as well as non-mass balance certified products, are put into the system. These are tracked, and the green credits allocation can go to the product that you're interested into. So thanks to the fact that we are really a big resin global buyer with the overall Berry Global, we have access to the 2 main technologies of mass balance. The first is ISCC Advanced Recycling, which is maximizing plastic recycled content. And the second one is the ISCC Biofeedstock, which is maximizing the CO2 emission reductions. So to make this a bit more visual and trying to relate this to infection prevention, okay, we worked on a case study for surgical drapes. Here you can see typical 2 or 3 layer structures for surgical drapes. And then let's see together what would happen if we would use 100% mass balance solution versus using 100% virgin fossil fuels raw materials. Thanks to our suppliers' data and to our estimation, we estimated that we can achieve up to 1,200 grams CO2 emission equivalent per a standard drape laminate. So if we try to think what would this mean, right, in practical situation, if you consider the surgeries per year in a medium-sized region in Europe, and you make, of course, estimations on the number of drapes used, which we underestimated to make some data, right, available for the discussion, you could save up to a town electricity use for 1 year. This means that if you move drapes to our mass balance solutions, and if we put together all the regions in Europe in the different countries, really think how powerful this could be. So again, these calculations, it's only made with estimations, which were taken really with caution, I would say, and only thinking about the drapes. So think about all the other products that can also be moved to this type of raw materials. So in conclusion, in the recent past, we saw that infection prevention needs a more resilient supply chain. And coming back to this, to the previous slides that I was sharing, I think that we are still in a very long wave of COVID, especially in the infection prevention segment. I'm not sure what will be coming into the region in terms of converting or not. I just think that this topic, it's on the table and people are continue discussing this due to the different challenges that COVID raised, like, right, for example, freight costs. In the present, you have different solutions for different needs, but I think we can see that there are major trends in infection prevention, which are, of course, the safety and protection, but this combined together with comfort. So with the healthcare worker also in mind, and keeping patients and user safety as priority number one. And then if we look into the sustainable future, really the healthcare supply chain needs to take action because these are global and especially European goals, so reduction of CO2 emissions. At Berry, we're committed to sustainability and want to support in this journey. And to this, the message is, if you want a safe and a sustainable product, you and your customers can get it. It's not fantasy. The technology is here, and we are happy to support in this. Thank you very much for your attention.
Do you want to leave now?
No.
No, okay. Thank you, Elena. Very nice. Now we'll have Daniele Restighini to come from Exton. He's responsible for product manager of medical applications and also member of the EDANA Infection Prevention Working Group. So welcome to talk about some other things in this area. I just want to make sure that in the middle you might have to leave. No, 10 minutes.
10, 15.
In 10, 15 minutes. Okay, sorry. So please go on.
Good morning, everybody. Thank you for being here. To good old friends and new people I meet for first time. Today, I'm trying to bring a point of view from a film manufacturer and laminate manufacturer. Exton is a Swiss-based industry in the film manufacturing, the casco extrusion, and makes laminates also for the medical business. What we are supporting today is that monolithic barrier films and laminates are preventing viral contamination. Starting from January 2020, we all know very well, down to September 21, in Europe, more than 47 million cases of COVID has been officially registered, and more, unfortunately, during these days. Worldwide, more than 230 million people are affected by COVID. And among these people suffering because of coronavirus, COVID-19, you have nurses, doctors, healthcare professionals, healthcare workers. In some European countries, they have been touched up to 14% of the health workforce. So during year 2020, and someone already mentioned this, many countries in Europe faced a lack of PPE, personal protective equipment. Not only face masks, but also gowns, coveralls, and many others, as you know. The European industry, and we are part of the European industry, and the plastic industry, we are part of the European plastic industry, had to react and find urgent solutions to this lack of materials, to support, to answer the healthcare professional request of PPEs and suitable apparels to face the pandemic. I am proud to say that the nonwovens industry and the plastic film industry, gave a great answer to this request of help from the healthcare workers. And we have been supporting this request in the best way with single-use materials. The nonwoven industry has provided polymer-based nonwovens materials suitable for the production of PPEs, and the film industry has used those nonwovens to be combined with barrier materials, both breathable and non-breathable. We have seen already some of the differences mentioned in some previous presentation. Those nonwovens have been laminated with films to support the production of personal protective equipment, resistant to virus penetration. For many years, we have been talking about resistance against virus penetration, and in some meetings, it was kind of theoretical discussion. But then, heard you know very well, then we came to reality, and we had people asking only and mainly for material resistance to viral penetration. With all the reference standards that we all know very well, all those standards that the film and nonwoven industry is taking care of together with professionals. And the resistance to virus penetration made X10 as a major European film producer to decide that there is one technical definition that sums up the key point. And the key point of barrier films and laminates, the word showing and indicating this key point is monolithic. We strongly believe that monolithic films, properly applied, properly extruded, properly used, better guarantee the needed protection to healthcare workers against viral penetration. The manufacturers of medical protective equipment, especially gowns and coveralls, are asking our industry, how can we reduce the risk of infection? This is a very old question, but it came back more and more. And additionally, they are asking our industry, nonwovens and films, and laminates, how can we improve the protection of medical health workers? The answer, and to answer these questions, we can compare briefly, because it has been already done in a previous presentation, compare the main properties of some existing solutions. Microporous film, monolithic non-breathable films, and monolithic breathable films. Microporous film, we all know that you have to stretch the material to get porous. It is breathable, but we believe it is not able to reduce the risk of pathogen penetration. So you have a microporous material which is breathable, but the level of protection can vary a lot. So you have breathability, but in this case, breathability represents a risk increase. We have then monolithic polyolefin films. They are mainly manufactured with low-density polyethylene and polypropylene. It's a perfect barrier. It's a perfect barrier. You can test it. We test it. We are doing many testing every day, every month, every year. It's a perfect barrier, but it's not breathable. So you have a lack of comfort. Again, this is very personal point of view. You have workers, they don't care about comfort. They want to be protected. So the main question is, again, how can we better protect the healthcare workers? The monolithic polyolefin film give the highest possible level of protection. But the workers have to forgo the comfort of breathability, which is something less important than protection. We have to keep this in mind. We have to remind that the first point, it's always protection. When you have a monolithic breathable film, we have seen also in a previous presentation, you have something giving, representing, guaranteeing a better pathogen barrier plus the breathability. You can see on your left that a monolithic film is able to stop the diffusion, to stop the virus going through. And if you are applying the right polymers, copolyester polymers, you have something which is chemically breathable. The water vapor is absorbed onto film surface and desorbed on the other film surface. With microporous materials, you have porous, you have apertures, you have a risk. So we believe that the diffusion of vapors represent, with microporous film, also a risk of diffusion of viruses. There is a cost of non-compliance. We are a little bit far from this because we are not manufacturing medical devices. We are producing materials to manufacture medical devices. But there is a cost of non-compliance because a PPE is a critical and essential safety instrument. During the last few years, the total value of class actions and settlement of PPE recalls and problems in the medical devices industry has a value of approximately $120 million. Difficult to estimate, but this is what we put together with the available information. What we believe is that instead of cutting costs, we have to improve the safety of healthcare workers, especially when talking about gowns and coveralls and face masks, but we are not in face mask business. Instead of cutting costs, let's work to improve the safety. Scientists developed a class of monolithic, breathable, thermoplastic copolyester. And these materials can be used for the extrusion of films combined with nonwovens to manufacture personal protective equipments with the highest level of protection against pathogen penetration. When monolithic films are properly applied, they are always positioned in the highest class of protection. Together with hot melt lamination, cast embossing co-extrusion can be applied to manufacture laminates for PP manufacturing. And both lamination technologies, hot melt or cast extrusion technology, offer the right protection level with diverse features. What I would like to deliver today as a representative of a film extrusion and laminates producer and as a member of EDANA is that we strongly believe and we continue to strongly believe in single-use solutions, which are also called single-patient solutions. We strongly believe in monolithic films to protect healthcare workers. And we believe that there should be no compromise between breathability and safety. Safety comes first. We are in the plastic industry, we are in the film extrusion, we are in the nonwoven business. We are not producing medical devices, but we have a great responsibility. We have to give our contribution, and our contribution to safety is based on monolithic films. When studying and developing solutions, our industry altogether, and we at Texton every day, we make the maximum effort to protect both patients and healthcare workers. This is part of our mission. And we believe that monolithic films and laminates properly made with monolithic films are part of the better word the nonwovens and film and laminates industry is creating and has created since a long time. Of course, together we have a lot further to go. But we have to keep in mind that safety, it's also our responsibility. Thank you very much.
Thank you, Daniele. And we keep the questions for later. And now we will also welcome John Birkett from nonwoven as a business director in medical. You will share something totally different, another aspect again on infection prevention. So you're welcome.
Good afternoon. Index 20, finally we're here. Reunited again, and what a pleasure it is to be here in person and meeting people and be out of the house. Just imagine for a moment, as we've already heard, if you get hospitalized for any reason whatsoever, you stand a very high chance of getting an infection. Healthcare-associated infections, or HAIs, are amongst the most common complications during hospital care. These infections can lead to significant morbidity and mortality, with, as we've heard again, millions of lives sadly lost every year, and are estimated to cost billions annually. Historically, such infections were long accepted by clinicians as an inevitable hazard of hospitalization. However, with the work of the groups here, it is now understood that relatively straightforward approaches can prevent many common HAIs. And as a result, hospitals and clinicians are prioritizing efforts to reduce the burden of these infections, and groups like ours today are focusing on what we as an industry can do to help. As a non-woven manufacturer, some of the tools we have to help aid this prevention measure are the utilization of controlled areas, which, whilst not being official clean rooms, are areas of the businesses where clean room style protocols and elevated levels of cleanliness are applied. In our case, our baseline manufacture, where the sheer scale of the equipment and assets makes siting in a clean room impractical. And of course, more specifically, that I'll touch on is our clean rooms themselves. Clean rooms are often required in the manufacture for many critical items, commonly used in the electronics industry for integrated circuits and hard drives, but also routinely used in the pharmaceutical and medical devices, where the environment may need to be free from bacteria, viruses, fungal spores, or other potential pathogens. So what is a clean room? Well, for those of you that don't know, well, to be exact, a clean room has a controlled level of contamination that is specified by the number of particles per cubic meter at any specified particle size. And for the purpose of this, I will refer to the classes specified by the International Organization for Standardization, headquartered right here in Geneva, set out in ISO 14644-1. Warning, this is my disclaimer alert. Other standards are available, and this does not represent any bias on behalf of the speaker. The last thing I want is a lawyer's letter from federal standards or something. These ISO classes are defined from 1 to 9, where class 9 is equivalent to room air and class 1 represents the cleanest possible clean room environment. As the classification numbers fall, clean rooms rapidly become cleaner and must comply with stricter standards. ISO 5 is the commonly accepted dividing line where clean rooms become more heavily restricted by rigorous requirements for filtration, contamination, and environmental control. So in simple terms, ISO 1 rooms are the cleanest and ISO 9 rooms the dirtiest, but still far cleaner than regular rooms. The majority of clean rooms fall within classes 7 and 8. We at Nonwoven Limited operate class 8 rooms, which are more than adequate for our needs. Typical types of contamination that need to be addressed include particulate contamination, and we've heard a little bit about this already, major sources of which can come from the room itself via dust and aerosols in the air, or from the operators working in the rooms via hair, skin, bacteria, clothing fibers and fingerprints and the such like, or from the equipment used in the rooms via flecks of processing chemicals and materials, dust, paint flakes, fibers, or dirty solvents. There are also chemical contamination. This generally occurs when unwanted chemicals get accidentally added to the desired processing materials. The amount that gets added is often very small or trace amounts, and under normal circumstances, this would be of minimal importance. And finally, there's biological contamination, a major source of which is bacteria. As we know, bacteria are a natural part of the environment and may act as either a chemical or a particulate contaminant. While a clean environment minimizes the number of bacteria that are initially in the air, this doesn't cover the potential operator contributions of bacteria to the clean environment. For example, a sneeze or cough will generally put both bacteria and aerosol particles into the air. Bacteria are on everyone's skin, and even a scratch will also place both skin flakes and bacteria into the air. Unwanted contaminants can infiltrate critical areas in a number of ways. Oops, excuse me. Particulates can enter by air, as we've heard, or on the feet of any carrier moving between the external environment and inside the critical area, examples of which could be rodents or insects. However, this is extremely rare with robust environmental measures in place. What are our sources of contamination? Well, the typical sources are often attributed in these proportions to these factors. However, studies show that contamination principally emerges from the people through skin flakes and oil, cosmetic, perfume, spittle, and clothing-related debris, or via the fomites, the walls, doors, handles, paint coatings, packaging materials used, room air vents, spills, and leaks. There is also fluids such as particulate matter, bacteria, moisture, floor coatings, cleaning chemicals, or the deionized water. Even the tools and machinery that are present in the clean room may generate contaminants due to the friction and wear, lubricants used, emissions and vibrations, and even the cleaning equipment, such as brooms and mops and dusters, can inadvertently contribute to contamination. And lastly, even the products themselves that are generated in the clean room may also inadvertently contribute to the contaminants. So how do we control those contaminants? The aim of contamination control is to permanently ensure a sufficient level of cleanliness in the controlled environments. This is accomplished by reducing or eradicating contamination to sustain an effective rate of production. Within Non-Woven Limited, this is particularly relevant to our controlled areas and our clean rooms. There are many preventative procedures in place within a clean room environment. These include training the clean room staff to strict standards in hygiene and operating practice. We also have incorporated an audible reminder which periodically tells the operatives to sanitize throughout the shift. And we also have a rigorous gowning procedure to prevent particulates from entering from the outside environment. Contaminants in the air are a major issue, as particulates and aerosols in the air do not behave in the same way as large particles. This is critical because the particles are very small and thus have large surface areas. The area is available for chemical reaction with other substances, as well as physical effects such as electrostatic attraction. We've heard that from some of the other presenters earlier also. For example, a large particle, say a paper clip, when dropped, simply drops to the floor. The situation for very small particles is different. They randomly move around and execute Brownian motion. They float in the air for a fairly long period of time. These particles in the air are removed via HEPA, high efficiency particle attenuation filters. These are typically 99.99% effective, and thus, at least in the absence of any new source of contamination, only the smallest of the small particles will remain in the air. These typically are removed by colliding with other particles until they are so large that they are removed by other means, or they stick to surfaces and are removed by cleaning in this manner. This is a bit like dust in a room where large aggregates of dust form dust bunnies that often hide beneath the furniture, while other dust simply rests and settles on top of the surfaces. Certain areas of the clean rooms have more stringent measures than others, such as the packaging areas, corridors, gowning rooms, transfer hatches, in order to maintain the clean room standards. Regular cleaning regimes are also implemented with thorough clean downs conducted between different jobs and additional controls applied to all the materials entering the clean room environment, such as double wrapping and bioburden testing. The main purpose of our clean room protocols within our quality management system at Non-Woven Limited is to ensure we run successful clean room manufacturing operations that provide repeatable safety and performance for all the products so that they meet the operational and user needs fully. We at Non-Woven Limited are very proud to work with some of the most recognizable names in both the non-wovens and healthcare industries, and truly believe for infection prevention that clean products are safer products. It's been my privilege to present today. I thank the organisers at Adana for giving me the opportunity. I look forward to, if there's any questions, I'll do my utmost to get those answered for you at the end. I wish you all well and would like to invite you to come and visit us if you have time at stand 1545. And I hope you enjoy the rest of Index 20 has to offer. Thank you very much for your time.
We have a few minutes left in the program for some questions. And please. Yes, please.
I'll keep my questions short because there are 4 of them. In case of a bioterror attack, that is, some jihadis planting a biobomb at the heart of Delhi, how ready are we? This is the first question. Should I list the 4 of them straight away?
Well, I don't.
Is that a question for me or?
I don't know who could
anyone.
answer.
It's probably beyond my pay grade.
I think we might need to leave that question open for us to discuss about and come back later on.
Okay, so
I think so. Yeah.
no one else wants.
But I have another question, though, and that is something I addressed with you, because I have met some people who are saying, when you wear a face mask, will there be some polymer, small molecules of plastic that go into your system, in your body, when you're breathing through a face mask? Is that really going to be a problem? And I addressed that with Gert Riesen.
Yeah. I knew that you would be asking that question, so yesterday evening I did some research and did some literature research, and I came to a publication from very recent, from China, and I did not finish the complete reading, to be honest, because normally I would have said that the risk is rather small, because you have different types of fibers in a mask, in any type of mask. You have different layers. Every mask has at least 3 layers, an outer cover, a filter, and an inner cover. And the inner cover, the filter is made of very small particles, microfibers, really microfibers. If you would place them here on your hand and you would inhale, you would definitely inhale them. But because of the fact that that filter is sandwiched in between 2 different layers of fibers, yeah, those fibers are actually kept in between those 2 layers. However, I found that publication from China recent, and that gave some very nice insights also in that question and in the answer, most probably. Yeah. First is that wherever we are, if we are outside, we are inhaling microplastics, because they are in the air around us. Yeah. So, and I'm not saying that we should not be worried, but we do inhale microplastics anyway, and most probably in big cities it will be more than on the countryside. And then what I've read, but again I have to finish reading, what I've read is that it is also depending on how you treat the mask, and for some people are even disinfecting a mask. I don't believe in that, but if you disinfect the mask, you will be more prone to inhalation of fibers.
Thank you so much.
You're welcome.
Yeah. Okay, and actually, unfortunately, you know, Professor Pitti had to leave because he's going to have this Clean Hospital Day now, and it's been it's a very good privilege to have him here and talk about the things. And I must say, we've covered so many different aspects of infection prevention, so it's really very interesting. So thank you all for your presentations. It's been very informative and interesting, I must say. And I think we are lacking a little bit of time. Is there one more question or so? Yeah, you do have. Yeah, it's it's Jason from in the US. I'm sorry, he might have some questions. Why don't you address a question then? He's willing to answer.
Absolutely. Good morning from the United States.
Yeah. Good morning. We have had a very good seminar here, very interesting. So, and your presentation was very good too. So thank you so much.
You bet.
Yeah. Okay. Any question? Now, I think we are running out of time, so I'm sorry for that. But, yeah. So you will get questions by the email or some the other way. Is that good for you?
That sounds great.
Super. Okay. Thank you so much for attending. Good,
Yeah,
good.
thanks.
Okay. Okay, thank you. Good. Thank you. Okay. We're done.
Since I
Yeah.
index, I am a journalist, and
I know, we've met in
I understand nothing. But
Okay.
I